Hello and welcome to Say That Again Slowly, the Cambridge Festival podcast where students at Cambridge University chat with the experts who have contributed to the festival. We try to pair up students with researchers and authors from very different disciplines to bring things back to basics. There are no stupid questions here. My name is Rebekah King. I'm a second-year PhD student in the Faculty of English Literature, studying the concept of dangerous magic on the early modern stage. Today, I'll be interviewing Selina Ballantyne, who is one of the organisers of an event called 3Rs, Animals in Biomedical Research, which will be on in the University of Cambridge Admissions Office, New Museums site, from 10 a.m. to 4 p.m. on Saturday the 2nd of April, and again from 12 p.m. to 4 p.m. on Sunday the 3rd of April. Thank you so much for coming to chat with me today. So Selina, can you introduce yourself and what your role is?
So I'm Selina Ballantyne. I work for the University of Cambridge. I work in the office for the Director of Welfare and Governance as a licence manager. Previous to that, I've worked in animal facilities for about 22 years, starting out as an animal care person all the way up to manager and leadership levels. So I've got a good background into what we're talking about today, so hopefully
yes,
it'll all go well.
Awesome, thank you so much. So what we're talking about today is you're doing a talk for the festival, is that right? And it's on the 3Rs?
Well, we have a stand at the festival, and it's
okay.
based around the 3Rs, and there'll be representatives from the university there over the course of the weekend to talk to members of the public and show them some of the stuff we do.
So it's going to be a stall and some interactive stuff at the stall.
Yeah, we're hoping
right.
so. We're planning to have things like isolators that people can actually have a play about with.
What's an isolator?
Oh, so an isolator is like a big bubble that holds animals, and it can either hold them for containment purposes or, but they could be really clean animals as well, which is what we use them for, and that keeps everything from our atmosphere out of their environment to keep them clean.
So you can actually sort of go inside it. This is like
you typically stand outside and put your arms through inside in some gloves and things, and then they're housed inside in their own microenvironment.
Oh, that sounds like fun. Okay, awesome. Thank you so much. So, so the stall and what you're talking about there is based around this principle of the 3Rs. Is that right?
That's right. Yeah.
And can you tell me what they are?
Yeah, in simplest terms, they're known as reduction, replacement, and refinement. When you work with animals in research, it doesn't matter what type of research it is, and it doesn't matter what position you hold. These are the three things that we try to adhere to and have at the forefront of our mind with whatever work we're doing, whether that's welfare and care or whether it's actually science-based. We're always trying to implement things to improve the lives of the animals, and in turn that actually helps to improve the data quality of the science that comes out.
Actually, that's a good point. I was going to ask you a question about that. So I was watching this video that the university had produced, and it said, it had this quote, good animal welfare and good science go hand in hand. So that sort of sounds like what you're saying there. Can you talk a bit more about how that works? Why does the animal welfare help the science?
Absolutely. So we want to produce really high level science, and to do that we have to reduce the variability, the data that you might get. A healthy, happy animal is always going to produce a better data set than one that's unhappy or unhealthy. If you think about ourselves, we always work and perform better when we're in a good place, when we feel happy, when we're motivated and things. We always perform much less well when we're demotivated, or we've got a cold, or we feel unwell, or something else is going on that's distracting us. And the principle's the same for the animals. So by promoting good welfare, we promote better scientific outputs.
Awesome, yeah. And I suppose there might also be things like stress chemicals and things that could get in the way.
Absolutely. Those types of things can create variabilities, depending on the research that you're looking at as well, on how that comes out in your data sets.
Great, so it's like a win-win if the animal is happy. Awesome. And I read up as well, and I found that apparently in the UK we've got some of the toughest legislation in the world, was the phrase that was used. So first of all, I was going to ask a bit more about what kind of legislation we have in the UK, and also whether you think anything needs to be updated or improved.
So I think it's fair to say we have quite a tough legislation, particularly when it comes to protecting animal welfare. But also allowing us to develop and create meaningful science. Our legislation is stringent in terms that it's also acceptable to other countries. So our permissive laws are adopted by many other countries, including the European Union, and their law is aligned very closely to our pre-established laws.
Oh, right, so they saw what we were doing and adopted it. Sort of started in the UK?
Yeah, our laws have been around since 1986, and in 2012 we implemented part of the EU directive into our law, and theirs is very strongly aligned with ours.
Oh, cool.
But we have to recognise as well that legislation for us is a societal opinion. When we look at how it's governed and things, that very much comes into play, and stakeholder interest does as well. Generally, the UK public is supportive of us using animals in research if it's for specific reasons, and those reasons are acceptable. And those acceptable reasons are things like medical and veterinary research, pharmaceutical industry and research. You know, if you think about what's been going on in the world, and developing vaccinations, therapeutics, medicines, pharma is a massive thing. But like I say, all of that has to be governed by the Animal Scientific Procedures Act 1986. And we're bound by that, and there's very many aspects to that, and very many responsibilities and people that have to take a pivotal role in making sure that our animals are protected at all times.
Excellent. Yeah, so that one act that covers all animal research in all the different genres, or are there sort of like slightly different versions of the act?
There are other acts as well that feed into it, depending what you're doing.
Right.
You've got the Animal Welfare Act that doesn't cover science, but there are aspects of that. So if we think about the Animal Welfare Act and 5 freedoms, we apply 5 freedoms to animals in science. There we have other bits of legislation, regulation, codes of practices, for example, that are built into ASPA, which is the Animal Scientific Act kind of acronym, that we have to work to as well, and that covers everything from lighting conditions, noise conditions, temperature, humidities, floor space, to make sure that if you've got a cage of mice, for example, they've all got a suitable amount of space to have their own space as well as being social. And that goes right through to dogs and non-human primates, etc. We've got the Veterinary Surgeons Act as well, because we have named veterinary surgeons that support the welfare of the animals, but obviously they have their own act that they have to adhere to. So it all feeds in together.
Gosh, there's a lot of laws, but it sounds like they're useful laws, and they're helping to ensure all sorts of good basic standards of care.
Oh, absolutely.
Awesome. In terms of anything needing to be updated or improved, what do you think about that? Is it pretty good what we've got at the moment?
My opinion is pretty good, but like I say, it's societal opinion plays a massive part. So as public opinion and morals change over time, we could expect that the public support for it might change as well, and that with that comes updates, improvements maybe. They might be tighter, tougher improvements, but we have to bear in mind as well that if that happens, there needs to be a balance between what we put in place and the ability to create practical science and actually continue to move forward and make improvements in medical health, because what we don't want is it to be so stringent that the science is impossible to create.
Sure, yeah, absolutely. So it's about that balance.
Yeah.
So what are some examples of times when we've been able to reduce or replace animals that have been used for research?
So one big thing that's out there at the moment, organoids, that and things like cell cultures and computational models are all great reductions in the amount of animals.
Sorry, what's an organoid?
It is I don't know how best to actually describe this. It's you take some cells from an organ and then you grow them a bit more into that organ. So
it's not an animal on its own. It's like but living cells.
Yeah. So if you take liver cells, for example, and then you can develop them into, you can divide them and grow them into
like a little liver blob.
Yeah, that type of thing. They're all great for reduction purposes. They allow us to do bits of work without actually having to then use animals to do it.
Very clever.
When we talk about replacement, though, it's the end game. It's something we're all striving to achieve, but actually there's very little, if anything, that you can use to replace animals at the moment. And that's because all the science that we do, particularly when it comes to medical research and veterinary research, you need to be able to base it on a living system to know if it's going to work before we're allowed to put into human trials. And that's governed by the Home Office. We're not allowed to do it and human trials until we've proved it works in a living system.
I see.
And at the moment, there just isn't that technology out there. We haven't yet developed something that recapitulates the human system in computational form or otherwise that allows us to go in depth and move away from animal use.
I didn't realise that you're actually not allowed to use. So I think sometimes people say, I'd volunteer to have a go, but you actually can't by law. You must test on animals first.
Yeah, we have to do this before we can then prove that actually we aren't going to, or as much as we can prove, we aren't going to harm human beings.
Yeah, absolutely.
Put it into them.
Yeah, that makes sense. I sort of asked this, and you kind of already answered this, but are there any more solutions on the way? So you sort of said, well, for now, it looks like actually replacing animals, I mean, that's very far off. We would need to somehow be able to replicate the complex systems that you get in a living thing. But are there any more maybe reduction solutions that you know of on the way?
We're always working towards them, and I don't doubt that they will come up. Something that's a bit more up and coming at the moment is the use of human cell culture systems. And this allows us to take cells out of human organs, and these could include stem cells, and that gives us the ability to genetically modify them. And once we can genetically modify them, it allows us to start studying new biologies, and it also gives us platforms to perform early drug discovery that we would have done in animals previously, but now they're working towards being able to do that without the use of animals.
So the genetic modification is actually a way of reducing the number of animals that you can use. That's really interesting. What was I going to ask? So when you said that, I had a little mini question as a response to that. Now it's just completely gone from my mind. Stem cells. So just remind me from biology at school. Stem cells are ones that can go into lots of different types, is that right? Yeah. So they're useful. If we extract stem cells, we can kind of make them into different kinds of cells, and then test
I don't know enough about the science to be honest, but I believe that's generally how it works.
That's just really useful for testing things on. Okay, cool. So human stem cells can be a viable alternative for animals. That's really interesting. I saw on the website, so mice in particular are being used to fight cancer, and is that happening at Cambridge in the Cambridge?
It happens in many research facilities, and Cambridge are also merged with CI, which is the Cancer Research Cambridge Institute as well. So we have a branch of that certainly. They're a great model because they help us to understand the mechanisms that underpin cancer in terms of how they grow and how they spread. And again, because mice can be genetically altered, it allows us to study the genetic causes of cancer, but their physiological aspect of them as well allows us to also study the reproduction of tumour types in the correct tissues and body systems as we would expect to see them in the humans. And now we're not obviously completely comparative in terms of mouse versus human, but it does give us realistic kind of views and perspectives of how it works to help us then turn these into new ways to develop and diagnose and treat the disease.
So presumably almost every kind of cancer treatment that we have has gone through, we have because of all, lots of mice and lots of animal testing. That's yes. Pretty much every single thing we'll have.
I can't think of one myself that hasn't. So
there you go. And why? So you mentioned about some of the qualities of mice that make them useful for this kind of research, but as you said, they're very different from humans. So why mice? Of all the other mammals and animals, why mice?
We always have to. We're governed by the law, and the law requires us to use the animals of least sentience that we can for the work that we need to do. That doesn't mean that there aren't studies using animals of higher sentience than mouse. So, for example, non-human primates, dogs, cats, they're all considered more sentient. We also have to think about the fact that the public are just more familiar with them when we think about dogs as companion animals. But mice have a lower level of sentience. But it's not just that. Mice are easily produced. They have multiple offspring in one litter. Their gestational period is very short. Their lifespan's only a couple of years. It allows us to then study the development of cancers and things through a lifetime rather than just a small window, which obviously with a human, you've got to wait many, many, many years to get to some stages of development of cancer.
I see, yeah.
The mouse, basically it's fast forwarded so that you can get there quicker, and because we can modify them and manipulate them as well, it allows us to put what constructs we want to study into a mouse. So you can either actually study it through induction, and that means manipulating the embryo with cell constructs and things, and then having it born and it is born and will have a genetic predisposition to the cancer, or we can implant cancers. So we can take some of those cells from a human cancer, for example, and implant it into a mouse and make a humanized model, and study it that way. So they're very versatile, but also actually really interestingly, mice aren't necessarily always for other studies the most used animal that we have. Mammal-wise, yes, but we have things like zebrafish and flies that are widely used as well for different parts of research, and we can do some research on them and actually then translate that into mammals later on if we need to.
And I was going to ask about with mice as well. So, you mentioned that you can have very many generations quite quickly. That means presumably you can also track generational inheritance of kind of cancer genes and things like that. I don't know if that kind of thing is going on.
Yeah? Yes, it does allow for that kind of traceability to occur, which again, if you're doing it with just humans, is, you can imagine how long it's going to take centuries to get meaningful data from it.
Sure, awesome. Okay, so those are the most used. Is it flies, zebrafish, mice?
Mice are the most used mammals. Zebrafish, amphibians, and flies. It's not always been around, but it's something that's used a lot for different types of research.
Interesting. What about flies? What are flies used for?
I am not familiar enough with them. Malaria studies, we certainly use them for that. But then we use them to help feed on mice and things as well. So there's a combination of the two there. So we can see how the disease translates into the mouse, but also what's going on with the flies.
Interesting. And I bet they have
mosquitoes even, flies.
Oh gosh, right.
Mosquitoes. Sorry.
Mosquitoes. Okay, yeah. So it's mosquitoes rather than flies.
No, really. Flies are used as well for other stuff. So it's just that they come in abundance. There's so many of them that we sometimes forget that they're there, and they're also used in large numbers in research.
Right, and I bet they reproduce really quickly and have a very quick lifespan.
Yes.
So again, same sorts of things.
Yeah.
Similar kind of considerations when you're picking which animals to use. Interesting. Okay, cool. And then are animals ever able to retire? So yeah, after they've been used, what happens to animals? Do they ever kind of go to a nice farm, or is that just no?
No,
too sentimental.
It's not sentimental at all. Yes, we can retire them depending on the species that we're using. We can certainly retire them. We have to adhere to legislation. Again, there's strict rules around it. It partly depends what they've been used for, and whether it's actually from the health and safety perspective.
Right, if it's a contagious disease, you don't want to.
No, exactly. But also, from our side, we rehomed into 2021. One of our facilities rehomed 58 animals, a mixture of guinea pigs and hamsters. But we have the legislation, but we also have our own vetting processes in place, which are quite stringent. So we're not just going to let these animals go off to anyone. It's very much like if you were getting a dog from a shelter or something, how they'd come and check you out. We do the same thing.
Awesome. So yeah, so some animals do get retired if it's safe and if there's a good home for them.
Yeah.
Then you sort of answered this one earlier because you said we're quite far off not ever needing to use animals in research. But do you think there will ever come a time?
It's like I said earlier, it's the goal, isn't it? It's kind of the dream to get to that point. I don't think it's possible to say that we absolutely will ever get to that point completely. But there's developments all the time and progression in science and things. And you think about organoids and things like that; they weren't around that long ago. They're relatively new. So there's always steps forward. So you never know. One day, maybe, but I don't think that it's not something we're going to see in the near future at all.
But this is the best way for now. The 3 R's is kind of the best, most responsible way of dealing with animals in research.
Yeah. Awesome. And we work to it all the time. I can't see this changing at all. We upholding the welfare of the animals only introduces good science, and that's what's best for everybody.
So there's a difference between animal technicians and the people doing the research, that's right?
Yes, yeah. So animal technicians are the guys that work on the ground floor with the cages and the animals themselves on a day-to-day basis, and they care for the animals. They look after the well-being, they do checks on them every day to make sure that they're in conditions that we expect them to be in. They feed them depending on the animal that you're working with. If it's a dog, for example, they'll play with them. Make sure that basically the welfare is upheld and they're happy and ready for the work that they need to do. But they also will work alongside the research scientists. So these are the people that are doing the science. And so they might come and do some of the actual experimental work themselves, but they might ask for support from welfare research technicians who will help. And obviously they monitor the animals after experiments have been done to watch their welfare, make sure they're not suffering anything that's unexpected or outside of the remit that's been allowed with the authorisation of the licence that allows the work to happen. So they work hand in hand, but there's a very significant difference. Technicians have a lot more emphasis on husbandry training and how to look after the animals. Researchers will be trained to a degree for handling and how to work with the animals to do their science and things, but really it's the people on the ground that have the hands-on work with them.
So presumably a lot of the 3R stuff that's kind of done by the researchers rather than the animal technicians, is that right?
It depends how you look at it, really.
Right.
It depends what the refinement is. If it's because you can have refinements anywhere from how you administer a drug, for example, through to how we actually care for the animals. And either way, if technicians are performing some of the administration, they will also have interactions with those types of refinements. But if you consider, for example, if we work with germ-free animals, it's just coming to the top of my head. I said earlier about isolators. So we would keep animals that are germ-free. So these animals don't have any gut microbiota. So they're very susceptible to anything environmentally. So they have to be kept really clean. And then what we would do is put something specific into them to make them notobiotic and then study that particular disease. But we would typically transport germ-free animals in an isolator. So if you think about a big square bubble that's got shelves and cages with lids on, but they aren't sealed lids because they need to be able to breathe.
Right.
And then you put it you want to move it from one facility to another. It's quite open. There's obviously we have ways to prevent movement and things like that, but there's always a risk. More recently, there's the introduction of a notobiotic germ-free isolation cage, which means that they can move in their own environment with a lid on and not have that risk or stress, and it's a refinement for them in that sense, and a refinement for us.
So a really practical solution to something that's good for the science and good for the animal, because it keeps stress levels down and it makes the environment more conducive to what's being done. And
technicians can have input into these types of things as well. In fact, there's some companies that work with us that promote this type of thing, and if you can come up with a product that's really good for refinement for welfare for animals, there's a competition and then they will make and sell the product.
Oh wow.
So there's one I'm thinking of is like a sombrero feeder thing to put things like supplementary food in for rodents, for example, so it's not in amongst their bedding and things, and it's easily accessible for them.
Oh, that's really cool. So can you talk a bit more about enrichment then? Because so one of the things that an animal technician might need to do is make sure that the animal's got stuff to do. They're not just in a kind of box. No. And they're having quite a happy life. So can you talk a bit more about that kind of thing? What sort of enrichment do animals get?
Well, it depends on the animal. Non-human primates, for example, if you're working with macaques, they live in harems, so they have a social structure. They have a foraging food on the ground. So what we're trying to do is allow them to exhibit natural behaviours, and that helps to enrich their environment. And if we go all the way to the other end, to mice, for example, they can have fun tunnels. They can have red houses. The way the cages are made, there's like a grid across the top that holds their food, but it allows for climbing behaviours and things as well. Nesting materials, because when they like to nest, they like to rip everything up and make a big nest themselves, and then they'll pull it all apart and start again. So we give them all of those types of things depending on the species and its requirements. we implement as much as we can do. So we make it as a natural environment as we possibly can whilst within a laboratory setting.
So it's similar, the kind of dynamic is a bit similar to, I guess, being a zookeeper versus being someone who works at a zoo doing research. So you've got a kind of an interaction, but one is a bit more hands-on and I guess practical, and then the other one's more sort of the academic side. I
think that's a good parallel.
And were you an animal technician, is that right?
I started as an animal technician all the way back in 1999. I've started out as a trainee, so coming in to do the training, all very new. I'd done at sixth form, I'd done an animal care diploma and MVQ, and then I just came straight into this industry. Trained right from the bottom, and whenever you train, all these elements of the 3 R's are built into it.
Was that there when you started, or is it fairly new?
The 3 R's have been around for a very long time.
Okay.
And it's ingrained into you as a technician. It's ingrained into you right from day one on how you handle your animals, how you treat your animals, how you look after them, and always thinking about, actually, can we improve this way or that way? So I started there and have come all the way up through kind of experimental, and then a welfare officer. So we have different people with different responsibilities under the law. And so now I've moved away to do more regulatory compliance work, because it's as important as any other aspect of the animal research, because it helps us to make sure that everything's been done correctly, and when adhering to the law, and by doing that we're making sure the animals are protected.
Amazing. You mentioned a little sombrero that someone had invented. Have you had a favourite improvement or refinement that you've seen over the years?
My favourite, and I don't know how well it's known actually, and it's probably my favourite because I was involved, we were doing containment work with parasites, and the way they would put them into our animals would be to have the animal kind of upright in a tube and their tail dipped into something for however long it took, and they would be conscious. We came up with a piece of equipment that basically allowed us to anaesthetise the animals short term and have them laid down all nice and warm and lovely, and their tails dipped into it down the bottom.
Like a spa.
So basically they went to sleep for a little while. They were exposed the way that we exposed them to these particular parasites, and then they come round and went back to the home cages, but they used to be held upright like this, and just with the tails dipped down whilst they're awake. So they're not very nice and a bit stressful and things, but now they don't really know what's happening.
Oh,
so
that's cool. I like that. I like the idea of them having a nice nap. Awesome. And then, so I didn't add this to my list, but you mentioned like the openness of research. I wondered whether you could talk a bit more about the principle of openness. It's a word that comes up a lot in the documentation that I've seen and about the event. So why is it important to do things like the event you're doing at the festival and chatting with me? Why is openness so important?
Well, if you think back, however many years, there seemed to be quite a lot of myth and secrecy around the research that we do, and of course that promotes distrust in the public, and we always will have groups that don't agree with what we do, but they very much had much more power over what the public heard and saw a long time ago. And the idea of openness is to reassure the public that we are doing everything that we can do to maintain the welfare of our animals, and show them how we conduct the science, and it's not barbaric or anything like that. That we are governed by laws, and we have to have relevant authorities in place for any piece of work. We can't just go and do whatever we like, whenever we like. And so there is a body that's been set up that's called the Concordat of Openness, for which we're signatories of, and many institutions that work in research have signed up to this. And so basically we have agreed that wherever we can, we will be open about the work that we do and how we conduct it. So we're answerable to the public in that sense. And you can see the trend change over time, and the more open we've become, the less issues there are with activists and things, and the more trust the public has, and that you can see that in how they've become more accepting of the fact that research needs to be done for medical reasons, but we all agree it shouldn't be done for cosmetics, for example. So
Yeah, that's really interesting. Yeah, and I think I saw some numbers. There are some numbers published in terms of openness. So it's things like how many animals are used, and there's some really good data that you can access. Anyone can access, right?
Yeah.
To do with what's actually going on, and also to I guess to kind of chart reductions, refinements, and all the things that we're talking about today. So there's quite a lot of openness in terms of just raw information, isn't there?
There is. So what the people responsible for doing the science, that hold the licenses that authorise them to conduct their work, they have to keep stringent records. And the reason they have to keep the stringent records is because they are required by law annually to return their figures for statistics, so it can be viewed. And these are published on the Home Office website, so we can see what numbers of animals are being used.
Even flies? Do they have to count the flies?
So it's important to understand that some animals, so we have to do this for protected animals, and when we call them protected, it just depends on what type of animal, if they're vertebrates, and what stage of development they're in.
Right.
So mammals, for example, are protected from 2/3 of the way through gestation, where a cephalopod is protected from the point where it hatches. And a fish from the point where it's capable of independent feeding. So if it predates in that point in their gestational cycle, for example, they're not necessarily protected. And if they're invertebrate, they're not necessarily protected.
Right. So they don't have to count how many flies are being bred and all that. That'd be a bit of a nightmare.
Exactly.
I think that was all my questions. I was sort of like just rambling at the end, just asking
That's okay.
extra random ones. Thank you so much for talking to me. Really, really interesting. If you're listening to this before the 2nd of April 2022, you can go and see Selina and ask more questions about the 3 Rs at the display which will be found in the University of Cambridge Admissions Office New Museum site from 10 a.m. to 4 p.m. on Saturday the 2nd of April, and again from 12 p.m. to 4 p.m. on Sunday the 3rd of April. Thanks for listening.